The next section of the nephron that we're going to focus on is the loop of Henle. So if we take a look at our nephron and we zoom in on this U-shaped structure, well, this will give us our loop of Henle. And the first thing to notice about the loop of Henle is that it's located inside the medulla portion of the kidney. So recall that the medulla portion is the lower portion of the kidney, while the cortex is the upper portion. So the upper portion contains the glomerulus, the Bowman's capsule, the proximal and the distal convoluted tubule, while the medulla portion, this section of the kidney, contains our loop of Henle as well as our collecting duct. Now, the second thing to notice about the loop of Henle is that around the loop of Henle, it contains a network of blood vessels, a network of capillaries known as the vasa recta. And the vasa recta functions to absorb the electrolytes and nutrients that are reabsorbed from the loop of Henle and back into our body. Now, the final thing to notice about the loop of Henle is that we can divide the loop of Henle into 3 sections. So as the filtrate travels down the tubule of the loop of Henle, this segment is known as the descending loop of Henle. At the bottom, the filtrate basically changes direction and begins to travel upward, and this segment is known as the thin ascending loop of Henle. The thin part simply means the size of our epithelial cells within this section are small. These cells are thin, they're relatively small. While this is our thick ascending loop of Henle, and thick means our size of the cells is relatively large. The epithelial cells are relatively thick. Now, the reason we divide the loop of Henle into these 3 segments is because each one of these sections carries out its own unique function. Now, before we examine the individual function of these 3 segments, let's discuss what the overall function of the loop of Henle is. So, the loop of Henle utilizes a system known as the countercurrent multiplier system. And what that system does is it increases the amount of solute, it increases the concentration of the ions found within our tissue surrounding our loop of Henle. And the tissue surrounding the loop of Henle is known as the interstitium. So it utilizes the countercurrent multiplier system to create a concentration gradient found inside the interstitium. And what that ultimately does is it basically allows our loop of Henle to absorb more water, and allows the nephron overall to absorb more water. And this concentrates our urine that is produced by the nephron, and at the same time, it utilizes as little energy as possible. So the only time we actually use any energy within our loop of Henle is within the thick ascending loop of Henle, and we'll see why energy is used in this segment in just a moment. So, what exactly is the meaning of the countercurrent multiplier system? Well, the countercurrent simply means that along the descending loop of Henle, the filtrate travels downward, but along our ascending loop of Henle, the filtrate travels upward. So we have a countercurrent. The 2 currents move in opposite direction. Now, multiplier means the loop of Henle multiplies the amount of water. It increases the amount of water that is passively absorbed back into our body from the loop of Henle, as well as from other parts of our nephron, such as the collecting duct. Now, to fully understand what the system is and how the loop of Henle actually creates this concentration gradient between the surrounding tissue and the lumen of our loop of Henle, let's begin with our thick ascending loop of Henle. So we're essentially going to work backwards. Now, as I mentioned earlier, the thick ascending loop of Henle contains epithelial cells that are relatively thick, relatively large. It contains large, simple squamous epithelial cells. Now, if we take a small section of our thick ascending loop of Henle, we basically get the following diagram. So these are the large, simple squamous epithelial cells. Now, as we see on the diagram, the membrane of these cells contains specialized proteins that are responsible for using ATP molecules, energy molecules, to establish a gradient inside our interstitium, between the interstitium and the inside, the lumen of our loop of Henle. So let's discuss how this is actually achieved. So let's begin on the basolateral side of our cell. The basolateral side of the cell is the side that points, that faces our basement membrane found in the interstitium of the surrounding thick loop of Henle. So this membrane contains sodium-potassium ATPase pumps, and these utilize ATP molecules, energy molecules, to basically move sodium ions against their electrochemical gradient. So 3 sodium ions are basically pumped from the inside of the cell to the outside of the cell. At the same time, 2 potassium ions are pumped from the outside to the inside of the cell. And what this ultimately does is it establishes a gradient for sodium. So inside the cell, we have less sodium than on the outside. And so what happens on the apical side of our cell, the apical side is the side that points towards the lumen of the thick loop of Henle, the inside of the tubule, we have these passive co-transport proteins that allow the movement of the ions down their electrochemical gradient. So because we established a sodium concentration gradient in which we have less sodium inside, the sodium will move down its electrochemical gradient from the outside to the inside of the cell. And at the same time, because this is a co-transport protein membrane, it will allow the movement of other ions as well. So for every 1 sodium that is moved inside the cell down the electrochemical gradient, our single potassium will be moved and 2 chlorides will also be moved. So let's take a look at the following diagram, which summarizes what takes place within the thick ascending loop of Henle. So what it does is it uses ATP to establish an electrochemical gradient that allows the movement of sodium, potassium, and chloride from the lumen of our cell, from the lumen of our thick ascending loop of Henle into these cells and eventually out of the basolateral side and into the interstitium of our loop of Henle, into the surrounding tissue. And this increases the solute concentration found in the interstitium. In fact, as we go down along the medulla, we have an increasing concentration of the solute of the sodium and chloride as we go lower and lower, and we'll see why that's important in just a moment. Now, between our cells, we have tight junctions, and these tight junctions prevent the leaking or the movement of water from the lumen and into our interstitium. So no water is actually absorbed and no water actually flows into our thick ascending loop of Henle. The thick loop of Henle is impermeable to water. Now, by the way, because we essentially increase the concentration of our interstitium found in the medulla of the kidney, what that means is we increase the osmotic pressure in the interstitium, and we'll see why that's important in just a moment. So let's move on to our descending loop of Henle, which is this section here. Now, at the top of our descending loop of Henle, the inside filtrate is isotonic with respect to our interstitium. Now, what exactly will happen as the filtrate travels downward? Now, the epithelial cells of the descending loop of Henle are permeable to water, so they allow the passive movement of water across the membrane of the descending loop of Henle. However, the cells are impermeable to ions, so the ions such as sodium and chloride do not flow across the membrane in the descending loop of Henle. Now, as the filtrate moves down, because the membrane of the descending loop of Henle is permeable of water, that means it will either flow out of the tubule or into the tubule. The question is, where exactly will it flow? Well, let's go back to the thick ascending loop of Henle. Recall that the thick ascending loop of Henle, what it basically did is it created a hypertonic environment in the surrounding tissue, in the interstitium. That means within the interstitium surrounding our descending loop of Henle, we have a high concentration of solute as compared to the inside. And because water always travels from a low solute concentration to a high solute concentration, water will passively move from the lumen, from the inside of the descending loop of Henle, and to the outside. So at the top, our filtrate is isotonic with respect to our surrounding tissue, but as the water continually flows out and out and out, eventually at the bottom of our descending loop of Henle, because so much water left our tubule, the inside of our tubule will become hypertonic with respect to the surrounding tissue, and this leads directly to the thin ascending loop of Henle. Now, before we go on to the thin ascending loop of Henle, let's mention the following things. So previously I said that the function of the loop of Henle is to ultimately allow the nephron to absorb more water. And it absorbs more water via passive diffusion without actually using any energy. So the thick ascending loop of Henle uses energy to establish an electrochemical gradient so that the water inside the descending loop of Henle flows out of the tubule without actually using any ATP molecules, and that only happens when the water moves down its gradient from a low solute concentration to a high solute concentration. Now let's move on to the thin ascending loop of Henle. So if we look at the following diagram, this is the thin ascending loop of Henle. And unlike our epithelial cells in the descending loop of Henle, the cells here are impermeable to water, but they are permeable to sodium and chloride ions. So the question is, in what direction will the sodium and chloride ions within the thin ascending loop of Henle actually travel? Well, recall that as the filtrate travels down the descending loop of Henle, the inside becomes more concentrated. So the relative amount of solutes inside our filtrate increases. And at the bottom of our loop, we have a hypertonic environment. Hypertonic with respect to our interstitium. We have a high concentration of sodium and chloride, and because our cells within our thin ascending loop of Henle allow the movement of sodium and chloride passively, these sodium and chloride ions will move down their electrochemical gradient from a hypertonic environment, from where we have a high concentration of solute, to a low where we have a lower concentration. So within the thin ascending loop of Henle, the chloride and sodium ions will once again move passively without using any energy to the interstitium. Now, as we'll see in the next lecture when we discuss our collecting duct, what the loop of Henle does is it increases the concentration of solute inside the surrounding tissue, and that allows water to be reabsorbed not only in the descending loop of Henle but also in the collecting duct, the final tubular portion of our nephron. So once again, let's summarize our discussion. So the loop of Henle is this U-shaped structure that is found inside the medulla portion of the kidney, and it uses a countercurrent multiplier system. It uses this system in which the filter travels downward here but upward here to multiply, to increase the amount of water that is reabsorbed back by our body from the filtrate. At the same time, it concentrates the urine in the collecting duct, and this basically is done without using too much energy. The only place we actually use energy is in the thick ascending loop of Henle. So within the thick ascending loop of Henle, we establish an electrochemical gradient by using ATP, and then in the descending loop of Henle, we utilize that electrochemical gradient to passively move water out of our tubule. And as a result of all that water moving out of our tubule, we concentrate our filtrate towards the bottom of the loop of Henle, and that's exactly why in the thin ascending loop of Henle, the sodium and chloride even further move to the outside, concentrating the outside even more. So this is the function of the loop of Henle. It ultimately concentrates the medulla, the interstitium found in the medulla of our kidney.